Hello everyone, welcome to Simplified Explanation. Today we will be discussing about types of antibodies. So in the last lecture we had seen the structure of antibody. The structure of the antibody was Y-shaped structure. It was a Y-shaped structure, and this antibody, it was containing two heavy chain and light chains. They were having two heavy chains and two light chains. So these were linked together with each other. The heavy chain and light chain were linked with each other with the help of disulfide bonds. And this region was the hinge region, right? This was the hinge region. And what was the function of the hinge region? It was providing the flexibility to the antibody. Flexibility to the antibody. Also, we had seen that it was the tip of the antibody was N-terminal and the tail of the antibody was C-terminal. Then we had seen that in the antibody structure there was a FAB region. And what was the function of the FAB region? It binds with the antigen, antigen binding site, binding with antigen, right? Then the lower portion was the FC region. And FC region, what it does, it interacts with the FC receptor of any effector cells so that it can produce an immune response. So yes, we know that it is having two heavy chains and two light chains. So the light chains, it can be either kappa or it can be either lambda, any one of the type. But the heavy chains, if we talk about the heavy chains, now based on the heavy chain constant region only, heavy chain constant region, the antibodies have been divided. The antibody, there are 5 types of antibodies. Okay, so the heavy chain is only the major part which is contributing to the types of antibody. It is having 5 types of antibodies. So let us now get to know what are the types of antibodies. If the heavy chain, if there is a gamma heavy chain, okay, if the antibody is having gamma heavy chain, then those type of antibodies will be known as IgG antibodies. And if the heavy chain is alpha heavy chain, then those type of antibody will be known as IgA. And if there is a mu heavy chain, then those antibodies will be known as IgM. IgE, and if there is a delta heavy chain, then those will be called as IgD. So there are 5 classifications of antibody: IgA, IgG, IgM, IgE, and IgD. So based on these differences, they all differ in their structural and functional properties. Structural and functional properties. So here there is a graph given over here, that is dictating that concentration of 5 types of antibodies in the blood. So here we can see which is abundant in the blood and which is the least abundant. So here we can see IgG is 75%, it's present in around 75%, IgM around 10%, IgA around 15%, IgD 0.5% and Ig 0.01%. These are not the exact values, they are only the approximate values given over here. So let's start with the first immunoglobulin, that is immunoglobulin G. So immunoglobulin G, if we say, these are the most abundant. Now only we have seen in the graph. These are the most abundant immunoglobulins in the serum, which constitutes around 80%, right? 80% or 75% of the total serum. These immunoglobulins, they are also most common type of immunoglobulin, common type of Ig that's found in the blood circulation. And when these immunoglobulin G are produced, when there is a secondary immunization, when there is a secondary immunization, these B cells, what they will do, the B cells will secrete IgG, most probably predominantly the IgG. So this was the brief about the IgG. Now let us talk about the structure of the IgG. Here we can see that's having this, the diagram is shown over here. The structure, if we talk, IgG structure is similar to the standard antibody structure that we have studied, but the difference here is that it's having 2 gamma heavy chain, right? That's why it's called IgG, and 2 light chain. Light chains can be either kappa or lambda, it doesn't matter, but the matter that matters is heavy chain, which should be gamma heavy chain. So here we can see this portion, gamma heavy chain is there, constant region, gamma constant region, then variable region. So this is the hinge region, okay, and they're having 2 disulfide bonds also. So let us start with the types. So these types, if we see, there are 4 human IgG subclasses, and they are distinguished by the differences in the gamma chains and gamma chain sequences, and they are numbered according to the decreasing average serum concentration. Means if I say IgG1, it is more abundant than IgG2, it's more abundant than IgG3, and then IgG4. So if we see, there is a decreasing average serum concentration. That's why they are numbered accordingly. Okay, so based on their constant region, let me erase this portion so based on their constant region, the size of the hinge region, and position of the interchain disulfide bonds between the heavy chains, these types have been classified. So although these subclasses, they are more than 90% identical on the amino acid level in these subclasses. So these subclasses, they have a unique profile with respect to antigen binding, immune complex formation, complement activation, triggering of effector cells, half-life, and placental transport. So here we can see the diagram also. The 4 types of IgGs has been shown here. That first IgG1, this is the IgG1, it's having 2 disulfide bonds. IgG2 having 4 disulfide bonds. IgG3, it's having 11 disulfide bonds. and IgG4 again 4 disulfide bonds. So based on different size of hinge regions and position of the disulfide bond, these IgGs have been classified. So next, if we see, let's move to next part, that is first type of the IgG is IgG1, right? So IgG1, as I have told you, that they are numbered according to the decreasing average serum concentration. So that means this is the most abundant IgG. This is the most abundant IgG. And it is important in mediating antibody responses against the viral pathogen. And how it does that? We know that we have studied in the antibody effector function that any antibody, what will it do? It will bind with the virus or virus particle with their Fab region. Okay. And then what they will do, they will after binding with this Fab region, the Fc region will bind with the any Fc receptor, and then it will activate the any effector mechanism. So further it will be activating the effector mechanism. Now IgG1, they can also, second or the third function of IgG1 is that they can bind with the C1q. Now what is C1q here? The C1q, it's a part or it's a component of the classical complement system, right? We had studied in the function part of the antibody, classical complement system, that suppose this is the IgG antibody, and this antibody, the Fab region has bound with the microbe. This is the antigen. This is the Fab region of IgG1, and this is the Fc region. So Fc region will bind with the C1q molecule of the classical component pathway, and then it will further initiate this complement. It will be initiating classical pathway, and after that further different steps will occur, and then MAC, a membrane attack complex, will form, and then there will be rupturing of the pathogen membrane. And then there will be the lysis of the pathogen. So in this way, this antibody is contributing, IgG1 is contributing in the lysis of the pathogen. So this is one of the another function of the IgG1. Now let me raise this portion so that we can have greater area. Now here we can see that's having the okay, so we were at the C1q, right? So C1q will bind with the Fc portion of IgG1, and further it will activate the classical pathway. Next is this Fc portion of the IgG1, it can interact with the Fc receptor of the effector cells also. And they can initiate or they can activate the ADCC, antibody dependent cell mediated cell cytotoxicity. We had seen this procedure also. So Then this IgG1, it acts as a model for the antibody therapeutics because it has some unique properties. What are those properties? It's having high thermostability, and we can see it's monomeric in nature. The structure, if we see, it's monomeric. And most important thing is that it's having average flexible hinge region. Which contains around 2 disulfide bonds. Right. So these are the properties of the IgG1 which makes it very useful for the antibody therapeutics. Now let us move to next type of IgG, that is IgG2. So IgG2, it plays a very important role, or we can say it recognizes or it responds against the bacterial capsular polysaccharide. That means this type of antibody reacts against they responds against the carbohydrate molecules. They will recognize antigen which are having carbohydrate on their surface or polysaccharides on their surface. And we can see it's having the disulfide bonds or a short hinge region which makes it less flexible also, right? So it is limited in its ability to activate the effector response. Like IgG1, it does not have that much ability to activate any effector mechanism or effector functions. So in the diagram we can see it's having short hinge region. It's around the hinge region. It's around 12 amino acid. Short hinge region, right? Short hinge region. And if it is very short and it's having another thing which is having 4 disulfide bonds, in the diagram we can see 4 disulfide bonds. So will it be able to be flexible? No, right? Because the short hinge region and also with the 4 disulfide bonds, it becomes very difficult for flexibility. They are restricting the conformational flexibility. It restricts the flexibility of the FAB arms. With respect to FC region. So that's why it's not that much flexible and therefore it's not able to activate effector response also. Next, moving to next type of IgG, that is IgG3. Now, as we know that it's present in a very small percentage of circulating IgG, and the main feature of this is that in the diagram we can see that it's having a long hinge region. Unique feature is that it's having long hinge region. In IgG2 we have seen that it was having short hinge region. It was having 4 disulfide bond. Here we can see in the diagram, let me show you, this is the hinge region, and they're having so much of disulfide bond, around 11 disulfide bonds are present, and also at the same time it's having long hinge region, right? So what from this what we can conclude? We can conclude that, first let me write the points of unique property of IgG3, long hinge region. And what is second unique point is it's having 11 disulfide bonds. 11 disulfide bonds. So from this what we can conclude? Again, it is just the opposite of that IgG2, right? It's long hinge region and it's 11 disulfide bond. So from here we can conclude that it's having greater flexibility, right? Greater flexibility. But what if it's having a long hinge region? Means that it is also having increased susceptibility to the degradation, right? Because it's having long hinge region whenever they are purified. So therefore these cannot be used as a therapeutic molecule because they can be easily degraded, because they are having long hinge region and they're having long hinge region, and therefore even though they are having greater flexibility, but at the same time they are having increased susceptibility to the degradation. So this was the unique property of IgG3. Then comes IgG4. Last type of the IgG is IgG4. Now IgG4, it's also, if we see the structure, it's also having short hinge region. And also they're having disulfide bonds. So the unique property, if we see here, is the unique property is half molecule exchange. That IgG4 can undergo half molecule exchange. They have the ability to undergo half molecule exchange. Also they can be termed as Fab arm exchange. Now what is half molecule exchange? The definition is been provided here. Half molecule exchange, the process by which two IgG4 molecules exchange a heavy chain, light chain unit to form a new IgG4 entity with specificity towards two different antigen. Now what does this mean? Let me explain you here. Suppose this is one of the IgG4 molecule. This is one IgG4 molecule, and it will undergo half molecule exchange with another IgG4 molecule. So this is another IgG4 molecule. And now they will undergo half molecule exchange. Now what will happen after half molecule exchange? One of the heavy chain and light chain of one of the IgG4, it will interact or it will combine with another IgG4, another half of the IgG4. So now they are having specificity for two different antigens. If I draw the diagram, let me change the color. Now they are having specificity for two different antigens. So this is what we call it as half molecule exchange, where the two IgG4 molecules, they're exchanging their heavy chain and light chain so that they can form a new IgG4. It's a new combination, no, because it's having specificity for two different antigen. Now this is showing the property of bispecific antigenicity. Bispecific, what is the meaning of bispecificity antigenicity? That it is specific for two different antigens. It can bind with the two different antigens. So while this unique property of IgG4 molecules confers anti-inflammatory properties also in nature, so therefore it's not desirable for the therapeutic antibodies. And also the allergens, one of the property of IgG4 is allergens are also a good inducers of IgG4. Along with which type of antibody are induced for allergens? IgE. Along with IgG4 are also induced. So this is one of the property of the IgG4, that it undergoes half molecule exchange. Now, the relative flexibility of Fab arms with respect to Fc region differs between the subclasses. Now we have seen all the structures of the IgG, their structures and the functions, their hinge regions and their disulfide bonds. So from here, how can we conclude the flexibility of the Fa arm, which is the most flexible IgGs which we have studied right now? We had seen that IgG3 was the most flexible. Then it was IgG1, then IgG4, they were not having that much flexibility, and last, the least flexible was IgG2. So in this order we can arrange the relative flexibility of the Fab arms with respect to Fc regions. So now we had discussed the IgG subclasses, right? So these subclasses were only of the human species. They were not the representative of all the species, okay? And in mammals, the number of the IgG subclasses, they can range from 1 to 7. So we can see that if one subclass is present, it is in rabbit. We had only seen the subclasses of humans, right? It will not represent all the species of mammals. In mammal, it is ranging. The IgG subclasses are ranging. Like 2 subclasses are present in sheep. 4 subclasses are present in cattle. 5 subclasses present in mouse. So all these are the subclasses which are ranging in different species. So next, moving to the comparison, this table is showing just the brief of whatever we have studied for comparing these antibody classes, IgG classes. Here we can see it has been shown IgG1, IgG2, IgG3 and IgG4. The general properties has been shown here, like molecular mass, it's shown in kilodalton, 146, 170, 146. Amino acids in hinge region, how many amino acids were present in IgG1? 15. 12 amino acid in IgG2. 62, the maximum one, which is making it most flexible, right? Then 12. Again in IgG4. So from here we can see that if there is a 62 amino acid in the hinge region, most flexible, then second flexible, then this one is third and this one is fourth. Inter heavy chain disulfide bonds, it's having 2 disulfide bond, 4 disulfide bond, 11 and again 2. Mean adult serum level gram per liter 6.98, 3.8, 0.51, 0.56. Relative abundance 60%, 32%, 4% and 4%. If we see the flexibility, segmental flexibility, it's good in IgG1, it's restricted in IgG2, very good in IgG3 and restricted in IgG4. Half-life 21, 21 and 7 to 21, 21. Placental transfer, it's good in IgG1. Again, it is also good in IgG2. IgG3 is also transferring in IgG4. Antibody response to proteins. IgG1 is showing, IgG2 is showing minimal response. This is also showing, IgG3, 4 are also showing. Polysaccharide, we have studied IgG2 was showing the most response against the polysaccharide. So yes, here it is showing the highest response. And IgG1 is lesser responsive to this. Minimal response by both IgG3 and IgG4. Then allergens, so which IgG was induced by the allergens, we had seen in the IgG4 subclass. So yes, this is the IgG4 subclass which is getting induced by allergens. It's showing maximum response. It's absent in IgG2 and IgG3. It's okay in the IgG1. And complement activation, C1q binding, every IgG1, 2 and 3 they will bind, but IgG4 will not be binding. So these are some of the classification, how we can brief them in a single table to understand in much simpler way. Now coming to the functions of these Igg type of antibody. So first, as I have told you, it will be, the first function is neutralization. So this antibody, they will be binding with any kind of viruses or toxin. As I have told you, it will bind with the Fab region, and then it will bind to the pathogen surface protein, and they will prevent these so that these virus cannot interact with the host cell. Okay. And further they cannot enter into the cell. So they are preventing these antibody, when they bind with the viruses, they prevent these viruses to enter into the host cell, and thus they are also inhibiting the replication of these pathogens. So also these Igg, they can also bind with viruses, they can bind with toxins, so that to prevent the infection. Second is opsonization of pathogen. So Igg binds to, and suppose this is the pathogen. Okay, this is the antigen, and it will, this Igg, what it will do, it will coat it. It will bind and it will coat these . Okay, so they will coat these pathogen, and after in this Fab region is interacting with the antigen, and this is the Fc portion. So Fc region will interact with the Fc receptor of any kind of phagocytic cells. Okay, this is the phagocytic cell, and further it will induce, so that there will be phagocytosis by the phagocytosis of the pathogen. Okay, it will mediate the ingestion by the phagocytic cell and pathogen destruction. So in this way, it is also used as opsonization. Activation of complement system, I have told you that it will bind with the C1q component. The C1q component, the Fc region of the antibody, it will interact with the C1q, this is the Fc region of antibody, this is the Fab region, and this is the antigen or the pathogen surface proteins, and it is interacting with this. Further, there will be activation of the initiation of the pathway, and after that MAC complex will form, and further there will be the lysis. So in this way it is also contributing in the lysis of the pathogen. Last is it crosses placenta. It can easily, these type of IgGs, they have the ability to cross the placenta, and the transfer of this IgG from the mother to the fetus, that transfer of the IgG from mother to fetus is a form of passive immunization. So this IgG is functioning or providing the passive immunization to the newborn, and this will be interacting with the neonatal receptor, means the neonatal Fc receptor, which is responsible for the placental transfer of IgG. So the important thing here is that it is providing passive immunization to the newborn. Okay, passive immunization. So 4 function we have studied: neutralization, opsonization, activation of complement system, crosses the placenta, and providing the passive immunization. Then now moving to the another class of immunoglobulin, that is immunoglobulin A. So it constitutes around 10 to 15% of immunoglobulin in the serum, and these IgAs, they are present in breast milk, saliva, tears, mucus of bronchial, genitourinary and digestive tracts. And if we talk about the structure, in which form they are present, predominantly monomeric in nature, but in secretions they are dimeric in nature. This is very important, that in secretions they are dimeric in nature. So structure, if we talk about the structure, the monomeric structure is the same as the standard antibody that we have studied. This is the IgA monomeric structure, monomeric IgA, right? It's having 2 heavy chain. What type of heavy chain? As I have told you, 2 alpha heavy chains, right? And 2 light chains, which can be either kappa or it can be lambda. Otherwise everything is same that we have studied. Moving to the another structure of antibody, that is IgA of an IgA antibody, is dimeric structure of IgA, that is secretory IgA. Secretory IgA. Secretory IgA is dimeric in structure. We can see. Now this dimer, if we see in the diagram, let me change the color. Now here, this is the diagram of the dimeric IgA. If we see closely, this part, this portion is the one monomeric IgA, and this another portion is another monomeric IgA, and they are linked. We can see this is the J chain It's shown over here, and this we see here is a secretory component. So now here from the diagram we can understand that this secretory IgA, it consists of two additional parts. What are they? J chain, and second is the secretory component. Secretory component. So let us talk about them one by one. First, let us discuss about the J chain. Okay, let me erase them. Now, J chain. First, J chain. So J chain, the two IgA monomers, they are linked to one molecule of J chain. Okay, the two IgA monomers, they are linked to J chain, which is around 15 kilodalton polypeptide. It's a polypeptide having a molecular weight of 15 kilodalton. And this J chain, it forms a disulfide bonds, right? What type of bond is formed? Forms disulfide bonds to both IgA monomers via the cysteine residue at the cysteine residue at C terminus. What I am saying here is that this is the J chain, and it is forming disulfide bond to both the IgA monomers with the help of the cysteine residues at the C terminus. Next, if we see another additional component of this structure, of this dimeric structure, is secretory component. So let's have a talk about the secretory component. So secretory component, it's 80 kilodalton, around 80 kilodalton polypeptide, and it comprises extracellular proteolytic fragment of receptor. Which receptor? I will be telling after a short time, but the name of the receptor is polymeric immunoglobulin receptor. It's small. PIGR, polymeric immunoglobulin receptor. And what is the function of this secretory component is that this is responsible for the transport of dimeric IgA or secretory IgA, dimeric IgA, where into secretions. From name only we can understand it will be transferring those IgA or dimeric IgA into the secretions. And so if we what we have seen, secretory component, it's 80 kilodalton polypeptide. It comprises the proteolytic fragment of receptor, that is PIGR, polymeric immunoglobulin receptor, and the function of this is used for transporting the dimeric IgA into the secretions. Now, if we see the structure again of this secretory component, we can get to know that it's having, let me first write, it's having 5 Ig-like domains. Ig-like domains that bind to Fc region of Iga dimer, and this interaction is stabilized by disulfide bonds. Disulfide bonds between where? Between the fifth domain of this secretory component and any one chain of the or any chain of the Iga. What I mean to say here is that if we see closely the structure of the secretory component, there are 5 Ig-like domain, like 1, this is 1, and this is 2nd, 3rd, 4th and 5th. So there are 5 in the structure. We can see there are 5 Ig-like domains are there, and where they're interacting with the Fc region of Ig. This portion is the Fab region. This is the Fab region, this is also Fab region, and this is also Fab region. This portion is the Fc region. So this secretory component is interacting with the Fc portion of the Iga. Okay. And how they are interacting? As I have told you, disulfide bonds, with the help of disulfide. And where these disulfide bonds are formed? With the fifth, this is the 5th one. If we see from here, and also this one is the 5th one. So between the 5th domain of the secretory component and any one of the chain of the Iga dimer. So in this way, we had understood the function of the Iga and also the structure of the Iga. Now, how these Iga, secretory Iga are formed. So, let us understand this with this diagram. This is the plasma cell. It's written over here. This is the plasma cell. So, this plasma cell, what they will do, they will start producing Iga. These are the Igas, right? And they will start producing Iga, and these Iga, then what they will do, they will migrate to the epithelial tissue. This is the epithelial cells. This is the epithelial cells, this is the submucosa, and this is the lumen. Now these Iga, they will start migrating towards the epithelial tissues, where these Iga, they will bind with the receptor. This is the receptor, and what I had told you, the receptor will be poly-Ig receptor. So this receptor is poly-Ig receptor, and these Iga, they will bind with these receptor. They are binding with this receptor, and where these receptors are present, poly-Ig receptor. They are expressed where? They are expressed on the basolateral surface. Basolateral surface of most mucosal epithelia, and on glandular epithelia in mammary, salivary and lacrimal glands. Now, after this, I was telling you about the plasma cells. They were releasing Iga, then after that they were migrating towards epithelial tissues, and then they will bind with these receptors. And after forming these, binding with these receptors, this complex, now it will be transported across the epithelial barrier to the lumen. Now this complex will be transferred from this epithelial barrier. This is the epithelial cells, and this is the lumen. So from here, the epithelial cells to the lumen, this complex will be transferred. And how this transport will occur? The transport will occur by it involves receptor mediated endocytosis. Receptor mediated endocytosis. Why? With the into the coated pits, and then we can see this, the vesicle is formed, so it's forming a vesicle. That's a receptor mediated endocytosis, and then they will be directed transport of the vesicle across the epithelia to the luminal membrane. This is the luminal membrane. Okay, so here it will be transferred or transported, where now this vesicle, what we can see, now this vesicle is fusing with this luminal membrane. This part, it has got fused with this luminal membrane, and after that what is happening here, it is written enzymatic cleavage. So the poly IgG receptor, this poly IgG receptor, it will be enzymatically cleaved from the membrane. Okay, and then becomes the, what will ultimately be formed, secretory component. Ultimately secretory component or secretory IgA will be formed, which is then released into the mucus secretions. So understood what is happening here. In brief, we can say that plasma cells are releasing IgA. They are binding with the receptor. These receptor transported by receptor mediated endocytosis. Then this vesicle will fuse with the luminal membrane, and after fusing with this, there will be the enzymatic cleavage of poly Ig receptor. Poly Ig receptor will be cleaved, and finally the Ig secretory component will form, and then they will be released into the secretion. So this is the way how the secretory IgA is formed. So what is the importance of the secretory component? Another importance here is that it masks the sites which are susceptible for proteolytic cleavage in the hinge region of the secretory IgA. It is masking the sites which are susceptible for proteolytic cleavage. So what is that process called? The whole process that we have seen here from the that is the transport of these antibodies across the epithelial layers into the lumen. That process is called as transcytosis. Okay, and this transcytosis is the feature of IgA. It's a feature of IgA. IgM can also transport against the across the epithelial layers. So, but most efficient is IgA. So the delivery of the antibodies to the mucosal surface of the respiratory, gastrointestinal, urogenital tracts, as well as its export to breast milk, requires the movement of the immunoglobulins across the epithelium. That process called as transcytosis. So the capacity of this transcytosis, it depends upon the properties of the constant region. Next is types. There are two types of IgA, IgA1 and IgA2. Moving to the functions. So secretory IgA, they are serving as an important effector function at the mucous membrane surfaces, which are the main entry sites of the most pathogenic organisms. And because it is polymeric, as we have seen, it's dimeric in nature, right? This is the diagram of the dimeric. It's dimeric in nature, so it can bind to multiple antigens. See, it is having so much of binding capacity. So it is polymeric. So secretory IgA, that can cross-link large antigens with multiple epitopes, and they can prevent the attachment of the pathogens to mucosal cells by binding to IgA to the surface antigens. And also the breast milk contains IgA, which helps protect the newborn against the infection during the first month of life. So this was all about the IgA and secretory IgA, their functions. Moving to next class of immunoglobulin, Immunoglobulin M. Now Immunoglobulin M constitutes about 5 to 10% of total IgG in serum. So it is having two first is first Ig. This is the first Ig immunoglobulin class produced in response to an antigen. So this is the first immunoglobulin class which is produced in primary response to an antigen. Second first is that it's first immunoglobulin to be synthesized by neonate. So it's the first class to be produced in response to primary response to an antigen. Also it's the first immunoglobulin to be synthesized by neonate. Now let us talk about the structure. Structure is, again, there are two types of structure of IgM: monomeric IgM. It is expressed as membrane bound antibody on the B cell receptor. Okay. Expressed as, let me write it over here, expressed as membrane bound antibody on B cell receptor, monomeric. And second is the pentameric form, pentameric IgM. It is these IgM, pentameric IgM, is secreted by the plasma cells as pentamer, in which we can see, let me show you the diagram first. This is the structure of the pentameric IgM and let us discuss it. See, this is the pentameric IgM and here we can see it will be in the secretions or in the secreted form, right? They will secreted by the plasma cells as pentamer. So here we can see there are 5 monomeric units. This is one monomeric unit, one IgM. Second, this is the third. This portion is the third. Then this is the fourth. This is the fifth. So we can see here what is 5 monomeric units are held together. And how they are held together? We can see this, they are held together by disulfide bonds. And then how these disulfide bonds are linking what? They are linking the carboxyl terminal heavy chain. This is the carboxyl terminal. We know that, no, that this is the N terminus, this is the N terminus, this is the carboxyl terminal. So they are linking the carboxyl terminal portion of the IgM monomeric units. So here we can write 5 monomeric unit held together by disulfide bonds, also where at the C terminus. And the 5 monomeric unit, if we see the diagram, they are arranged with their FC region in the center. This is the FC region again, and this is the FAB region. So all the FC region, this is the FC region, they all are concentrated. This is the FC region, FC. They are all present at the center. They are towards the center of the pentamer. And the antigen binding site, how many antigen binding sites are present? Let me first erase this so we can have a clear understanding. So here we can get to know how many antigen binding sites are there. 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. There are 10 antigen binding sites. 10 antigen binding sites on this antigen binding on these FAB regions, no? And if we see the diagram, there is additional, okay, let me change the color. There is an additional FC linked polypeptide. This I am talking about this chain. What is this? This is a J chain. This is the J chain, and it is binding to which portion? Again, it is linking the FC region. Okay, so J chain is linking the FC region of the IgM molecule, and this, as we know that J chain, they will be involved in disulfide bond to the carboxyl terminus. So these J chains, they are required for polymerization of these monomeric IgMs, so that they can form pentameric IgM, and it is added just before the secretions of the pentamer. This is added just before secretion of the pentamer. So we have seen what is the valency. We had seen the valency now only. If we talk about the valency, what is the valency? It's having 10. Because it's having 10 binding sites. It's the highest valency that any kind of isotypes. Moving to the functions of the IgM, it neutralizes viral infectivity. It is activating complement pathway. It can cross the epithelial lining to enter external secretions that bath mucosal surfaces. And the presence, this is because of the presence of the J chain. No, this in the crossing of the IgM across the epithelial lining is due to the J chain, because it's having J chain. No, that's why. Next class of the immunoglobulin is IgE. Now this is present in a very low concentration. Structure, we know this is the standard structure of the IgE, but it's having heavy chain. Otherwise, here we can see there's a standard structure of IgE. Now the function, let us talk about the function of the IgE. IgE, first, okay. So Ig antibodies, they will mediate hypersensitivity reactions that are responsible for asthma and anaphylactic shocks. So here what is happening, this is the Ig molecule, this is the Ig, and it is binding to the Fc receptors on the membrane of, this is the Fc receptor specific word. Which cell is this? This is the mast cell. Also it can bind to basophil cells also. So they will bind with the receptor, and this Ig molecule, when it is cross-linking with the allergen, this is the allergen. Okay. So the cross-linkage of this receptor, this is the receptor bound, so cross-linking of the receptor bound Ig molecule by the antigen or the allergen, what it will do? It will induce the basophils or the mast cells, so that they can translocate their granules. These are the granules. It's having many granules inside them, the mast cells or the basophil cells, and they will induce, after binding with the allergen, these IgE, they will induce the mast cell or the basophil cells, so that they can translocate their granules to the plasma membrane. Okay, and then what will happen, these granules, when they will, and suppose these are the granules, so they will be allowed to move towards the plasma membrane. Okay, and when they will move, So they will be allowed to, or they will induce these granules to move to the plasma membrane, and there they will release the contents. After moving there, they will release the contents to the extracellular environment, and this process of releasing or bursting of the granules is called degranulation. So here we can see degranulation, release of granule contents. As a result, active mediators are released. So histamine and other substances that mediate allergic reactions. So here we can see this degranulation is happening over here. Okay. So this was all about the function of the IgE. Last, immunoglobulin type D. So it's around 0.2% in the blood, and IgD along with the IgM, it is a major membrane bound immunoglobulin. Major membrane bound immunoglobulin. So its function has not been identified till yet. So this is the structure of IgD. It's having heavy chain, which is delta heavy chain. So let us summarize all of the things that we have studied, the types, their functions, their properties in a single table. Here the property, the classes have been given: IgG1, G2, G3, G4, IgA1, IgA2, IgM, IgE and IgD. So molecular weight is given over here. Heavy chain component, what will be the gamma 1, gamma 2, gamma 3, gamma 4. In A, alpha 1 and alpha 2. In M, IgM, mu will be there, and in D, delta will be there. Normal serum concentration 931. It's given all over here. In vivo serum half-life in days, 23 days, 23 days, 8 days, 23 days, and so on. Classical pathway activation. So Ig1, it is activating, minimal by IgG2, good by the IgG3, not by the IgG4. Also IgA2A1 they are not showing, high is shown by the IgGM, and again but not by the IgGE and IgD. Then crosses placenta, it's feature of all the IgG classes. So they are all are crossing, others are not. Present on the membrane, so which is the feature? As I've told you, M and D will only be present in the membrane, all will be absent. Binds to Fc receptor, only the IgG class will bind, others not. Mucosal transport, which will be those? It's a property of IgA and IgM. And induced mast cell degranulation, only IgE. So this was all about the types, their functions, their structure, properties of the immunoglobulin classes. So thank you